Hello and welcome to another session of Physics Wala where we are going to discuss about semi-autonomous cell organelle. So I hope all of you students are doing great and you guys are healthy and fine. So today we are going to talk about semi-autonomous cell organelle. So without doing further ado, let's start and discuss this. Great. So we are going to talk about semi-autonomous cell organelle. So in this category, we are going to talk about two of the most important cell organelle of eukaryotic cell, where we see they are not completely dependent on cell, means they are not completely under the regulation of nucleus. What we see on the other hand, they go in a partnership or they go hand in hand in an experience with nucleus and their own setup. So we see that semi-autonomous cell organelle, they are not under complete control of nucleus. So we have to understand on which parameters we will gonna classify this cell organelle as semi-autonomous. So what we see that these cell organelle have their own genetic material. So we see that they have their own genetic material. So beside the genetic material, we see they have independent setup for transcription and translation. So we know transcription is RNA synthesis and translation is protein synthesis. So what we see happening inside this cell organelle are three of the major incident of central dogma, where we see that they have their own genetic material, so they can take part in DNA synthesis, or what we called as replication. We see RNA synthesis or transcription. And we see protein synthesis or translation, right? So we see that in an eukaryotic cell, replication or DNA synthesis occurs generally in nucleus for the nuclear DNA or the main genetic material of a cell. But on the other hand, if we talk about semi-autonomous cell organelle, there also we see that replication of their own genetic material is happening inside these cell organelle. Beside that, we know for majority of the RNA of a cell, eukaryotic cell, The RNA synthesis takes place inside the nucleus. But here for these semi-autonomous cell organelle, where we see that transcription or RNA synthesis do take place inside them as well. We see that they also have a protein synthesis machinery. So which means if the protein synthesis also has to take place inside these cell organelle, they must possess ribosomes, right? So we see that point number 5, please jot it down. They have ribosomes. Now, what we have to keep in mind that these ribosomes which are present inside the semi-autonomous cell organelle, these ribosomes are basically 70S type of ribosome. So this we have studied, and probably this is going to be one of the very important point here, that 70S type of ribosome, they are not only present in prokaryotic cell. 70S type of ribosome, they are also present in the cytoplasm of eukaryotic cell, but not truly in the cytoplasm. They are present inside the semi-autonomous cell organelles. So we see that when we talk about the presence of protein synthesis machinery, we see these organelle also have 3 different types of RNA. So we see that this type of cell organelle, they have their own tRNA, they have their own rRNA, and they have their own mRNA. Okay, they have their separate setup altogether, what we can see. Now, if we talk more regarding this, we see their genetic material is circular. Okay, their genetic material is circular and guanine cytosine rich. These are the 2 very important points what we have to remember, because we are going to talk about another theory associated with semi-autonomous cell organelle, where we will see the significance of these points. So please mark these points which are going to benefit you in further learning, that they have 70S type of ribosome, their genetic material is circular and GC rich, they are not under complete control of nucleus, what we have to remember. And these are the couple of points what we should remember, that they are having circular and GC rich type of genetic material, they have 70S type of ribosomes, etc. Now, next we are going to talk about 2 more very important points regarding them, where we see that they are autonomously duplicating in nature, which means if these cell organelles has to increase in number, they can duplicate and increase in number in the cytoplasm of the eukaryotic cell, and that duplication process is not under the control of nucleus. So we see that there is a partnership running, so it's not completely dependent on nucleus. So we see this autonomously duplicating nature is one of the proof for that. We will now so obviously mark this statement as well, one of the important statement from these topics. What we will see again, that one of the common features for semi-autonomous cell organelles is that they are double membrane bound. Okay, so we see that they are double membrane bound. Again, this is also a common feature of these cell organelles. Now, without doing further delay, let us talk about who are these semi-autonomous cell organelles. So we see mitochondria and plastids are both comes under the category of semi-autonomous cell organelle. So we see that these two are going to be our center of attraction. We are going to talk a lot about mitochondria and plastid. So we see in both the cases, they are double membrane bound, correct? We see that they have their own genetic material, which is circular DNA, double-stranded circular DNA, and again having a very high GC content or guanine cytosine content. They have their own transcription setup. So we see that they have, three of the different types of RNA which are present in both of them. So they have their own tRNA, own mRNA, own rRNA. We see that in both the cases, inside the membrane-bound matrix, there is the presence of 70S type of ribosomes in both the cases. And also we see that in both the cases, they are, self-duplicating in nature. So they do not need a permission from the nucleus to duplicate themselves. Now, what we are going to talk about next is another relation with semi-autonomous cell organelle, and that is known as endosymbiotic theory. So before we go, please mark mitochondria and plastid here because they are the superheroes. So we see, please write this point here, that semi-autonomous cell organelle are also endosymbiotic in nature. Now we have to talk and understand what does that even mean. What is endosymbiotic mean? So we see that this is a cellular evolutionary theory which is known as endosymbiotic theory. So let us talk about it. Endosymbiotic theory this is a cellular evolutionary theory cellular evolutionary theory, where we see this theory claims that the origin of mitochondria and plastids are basically prokaryotic. So what exactly this theory is claiming? This theory is claiming that mitochondria and plastid were previously basically bacteria. Okay, so what we see that this theory talks about the prokaryotic origin of mitochondria and plastids. Okay, so it talks about the prokaryotic origin. Now we see, before we understand the endosymbiotic theory in a detailed manner, let us talk about what this theory is trying to say in brief. Okay, so we see that this theory talks about the presence of a ancient cell. So this presence of an ancient cell, this is often known as protocell, or we call it progenote. So we see, if we talk about protocell or progenote, this cell particularly claimed to be the first evolutionary cell. So it is not considered to be either prokaryotic, or it is not also completely eukaryotic. So we consider this as protocell or progenote, which is, going to be the mother of prokaryotic cell as well as the mother of eukaryotic cell. Now what we see that they have a cell membrane structure, and they have the genetic material inside. Now what we see in the course of evolution, what we see that the cell membrane of this protocell or progenote is going to fold and create a membrane for the genetic material which is present inside the cell. So what is happening here? We see that the cell membrane has infolded, and not only it has infolded, it has now created a covering for the genetic material which is present inside this cell. Can we see that now? So we see that this genetic material which were present, so now they have a covering, and this covering is, this covering is now known as the nuclear membrane. Once a cell has formed its nuclear membrane, henceforth we are going to see that this cell which is having a covering for its genetic material is going to be the progenitor for eukaryotic cells. Or you can say, from this point we are going to call it an eukaryotic cell. So what we see that once the nuclear membrane has formed, you know that this nuclear membrane is working as a origin for endoplasmic reticulum. So this we have studied, guys, earlier, right, in endomembrane system. So here we see that this nuclear membrane is working as an origin for endoplasmic reticulum. And slowly we see, due to the connected origin, we see that endoplasmic reticulum will give rise to Golgi body. And from Golgi body, you see that it can produce lysosome. So what we see here, we see now we have a eukaryotic cell with endomembrane system. So this is exactly what we have prepared. We have prepared an eukaryotic cell with endomembrane system. Are we understanding this or not, guys? So we see that this is the cell which is having the nuclear envelope, and we know, as we have studied already in the endomembrane system, that outer nuclear membrane works as an origin for rough endoplasmic reticulum. Rough endoplasmic reticulum produces smooth endoplasmic reticulum. From there, smooth endoplasmic reticulum produces Golgi body. Golgi body has Golgian vesicles, which develops into lysosome. So we see that this GERL network is already there in this eukaryotic cell. Now the question is, what will going to happen next? And that's what we are going to see. So now we are looking at a cell. This cell which is having its nuclear envelope, and you know that this outer nuclear membrane, this extends and give rise to endoplasmic reticulum. ये आपने last class में पढ़ा है, so that you have to keep in mind. So this we see is endoplasmic reticulum. We see the presence of the genetic material which is lying inside the nucleus. Now what we see here, that this cell is having the nucleus as well as the GERL network. So nucleus plus GERL network. If this cell tries to engulf a bacteria, now this is very funny, right? So it is trying to engulf a gram-negative aerobic bacteria. So must be the cell was hungry or something, and the cell was trying to engulf this bacteria as a food particle, and that was the moment the story begins. So we see that it is trying to engulf the bacteria by the process of course, we know this is phagocytosis process. So once the engulfment is done, once the engulfment is complete, what exactly we are going to see here? We see that, let's imagine this is the nucleus and this is the genetic material what they have, and they have now created a phagosome, and inside this phagosome you have the bacteria. Okay, so this was the bacteria. Obviously, please mark the origin. This portion is important. So we see that now you have a phagosome, and inside the phagosome you have the bacteria. Now this phagosome for some reason, uh, for some reason remain undigested inside cell. Now this is the story begins. So what exactly happens? So we see that the cell has engulfed the bacteria, and the bacteria couldn't able to get digested further. So we know this, we have studied this before, right, while discussing about lysosome. So we know that once the phagosome enters, it's supposed to fuse with lysosome. So something has happened with this cell, and it couldn't able to fuse its lysosome with the phagosome, and this phagosome, which is now containing this live bacteria, it remained undigested in the system. And the moment it remained undigested, we see that this bacteria has started a symbiosis process with the cell itself. It starts a symbiosis process with the cell. So once it started the symbiosis, the mutualism, we see that cell now do not want to kill it. And at the same time, this gram-negative aerobic bacteria has started growing its number by division. So we see the next step, in the next step, what we see that you have this eukaryotic cell where we have nucleus and its genetic material and etc., is now having more of these membrane bound bacteria which are under endosymbiosis with the cell. So can you see that that particular bacteria, which was initially just one, has divided and increases its number. So we see that it has increases its number by division, and now they are under a full-blown mutualism with the cell. They're under a complete mutualism. But the mutualism is happening, or the symbiosis is happening inside the cell. As this is happening inside the cell, we call it endosymbiosis. So we see that these bacteria, okay, they are now evolved as mitochondria. The moment you will say that it's a eukaryotic cell which is now carrying mitochondria, what we will see is this cell is going to be the progenitor of animal cell. This is going to be the progenitor of animal cells. So we see that from this cell in the evolution, later on, animal cells will be produced. So this is basically, in a way, the grandmother of all animal cells, what we have. Now the story doesn't end here. The story doesn't end here because we have only talked about mitochondria, and we are now going to talk about the plastids. So what we see is basically an extension of the previous story, where we see that a cell, a progenitor of animal cell, rather we should say like that, where it is already having the nucleus, which is carrying the genetic material, of course, and it is having its own, the endomembrane system, and we see that it is carrying now the mitochondria. This cell is trying to engulf another bacteria as food product. So you see that it's still hungry. So it's trying to engulf a bacteria as food. Okay. Now, what type of bacteria is this one? That we need to know. So, if you talk about this, this is a gram-negative facultative anaerobic. Facultative means temporary. Facultative anaerobic photosynthetic bacteria. So we see now the trouble. So we see that this bacteria, the progenitor of the animal cells, which it's trying to engulf, it's basically a gram-negative bacteria. It's a facultative anaerobic bacteria, which means it is temporarily anaerobic. Generally, it's aerobic, but sometimes if it requires, the bacteria can switch their mode of respiration from aerobic respiration to anaerobic respiration. So it can be facultative anaerobe. Now, and at the last point and the important point is that it's also a photosynthetic bacteria. So this progenitor of animal cell, it is trying to engulf this bacteria, which is obviously having these three features, and again the same thing happened. Means that the bacteria has successfully engulfed it but couldn't able to digest it. So we see that the nucleus and the genetic material, we see the mitochondria are here, right? These are the mitochondria. And we see that they have now engulfed this particular bacteria, which is photosynthetic, gram-negative and facultative anaerobic in nature, and remain undigested. So this also remain undigested inside cell. It remains undigested inside cell. And what happens when it remains undigested? It goes under endosymbiosis. So we see it directly goes under endosymbiosis, just like the previous case, and it evolved inside the cell as chloroplast. Okay, so this thing we have to keep in mind. Again, we are going to make the story a little more detailed here. So we see that the chloroplast is basically again a bacteria which is gram-negative, facultative, anaerobic and photosynthetic, which has been engulfed by this progenitor of animal cells and remain undigested inside the cytoplasm, and then it has gone under endosymbiosis and evolved as chloroplast. So now the kind of cell we are looking at, because this undigested photosynthetic bacteria can also divide in the cytoplasm and it will also increase their number. So basically you will going to produce something like this, where the nucleus is there, where the mitochondria are there, and also there are these chloroplasts. So we see these chloroplasts are there and these mitochondria are there. So both mitochondria and chloroplast, when they present together, we see this is the progenitor of plant cells. So we see they can carry plastids and mitochondria both. Isn't it interesting? So please mark these points which are important and can be asked as a question here. So I think a couple of things are being very clear from this, that why the semi-autonomous cell organelle are having so many common features, that both of them are double membrane bound, both of them having their own genetic material because they are having a bacterial origin or a prokaryotic origin, why they are having high guanine cytosine content in their genetic material, because we know that prokaryotic genetic material are having a high GC content, right? So that you get to see over here. We see that they have their own, ribosome, because we know that in prokaryotic cell there is the presence of 70s type of ribosome. There is the presence of the structure of polysome, what you can see also inside these cell organelle, which has basically having a prokaryotic origin. So this is all about the semi-autonomous cell organelle and the endosymbiotic theory, and what is the connection between semi-autonomous cell organelle and endosymbiotic theory. What we are going to talk about next is we are going to discuss both of these two cell organelle separately so that we can have even a more clear and detailed picture. So let us first start our discussion with mitochondria. So this is the first double membrane bound cell organelle what we are going to discuss in this chapter. So we see the first point, you write that they are double membrane bound. They are double membrane bound cell organelle. Point number two, as we are talking about the double membrane, we need to know how many membranes are there and what is the architecture or the organization of these membranes. So we see that there is the presence of a plain, sorry, there is the presence of a plain outer mitochondrial membrane. Okay, and there is no infolding present as such in this membrane. So this is a outer mitochondrial membrane, and then we have the presence of highly convoluted and, or you can say that highly infolded inner mitochondrial membrane structure. So we see that the space which is present between the inner and outer mitochondrial membrane, the space which is present here, this particular space, this is known as perimitochondrial space, perimitochondrial space, or we can also call it the intermembrane space. Inter means between two of the membranes. That's why this is also known as intermembrane space, or we can also call it the outer compartment. Sorry. Okay, so these many names are there for this area which is present between outer and inner mitochondrial membrane. Now, if we go and talk in detail about outer mitochondrial membrane, which is also known as OMM, and the inner mitochondrial membrane, which is also further known as IMM, what we see is that outer mitochondrial membrane, or OMM, are having a high rate of permeability. So they are having a high rate of permeability. That does not mean that it is semi-permeable membrane or it is a permeable membrane, nothing of that sort. They are still selectively permeable in nature. The only thing which has changed, that we see that there is now an increase of permeability rate, so that we can see with outer mitochondrial membrane. So we see that outer mitochondrial membrane, they are having around 40% lipid and 60% protein. Okay, so they're having 40% lipid and 60% protein. On the other hand, if we talk about IMM, or inner mitochondrial membrane, we see that inner mitochondrial membrane is exactly the opposite. That means having a low rate of permeability. Low rate of permeability. Again, when we see that it is having a low rate of permeability, does not necessarily means that it's an impermeable membrane. It's still selectively permeable, because we know that all living membranes are selectively permeable in nature. So it's still selectively permeable in nature, but it is having a low rate of permeability. So what we see composition wise, it is having 20% lipid and 80% protein. Okay. Now we are going to talk a little bit more about the inner membrane specifically, because this inner membrane is very interesting. It is infolded. So we see that the infolding of inner membrane, this specific area, if we look, the infolding of inner membrane helps to increase the surface area, right? So if there are more infoldings, that means more surface area. Now, all of a sudden, why we are so concerned about surface area? what is happening if we even increase the surface area. So we see that the inner membrane, it basically works as a base to carry a lot of enzymes which are membrane bound. So in this case, we see that if you have more enfoldings, that means you have more surface area. That means we have now more space to basically keep these membrane bound enzymes, right? So these infoldings, I guess everybody, those who have studied studied biology in 9th and 10th, they know that these are known as Cristae. These infoldings, specific infoldings, they are known as Cristae. So we see that the inner membrane of mitochondria carry membrane bound proteins. Two of these specific proteins we would like to discuss here. So the first one we are going to talk about is succinate dehydrogenase. So this succinate dehydrogenase, this is very important in terms of our examination as well. Frequently asked question, I can say that this particular enzyme, this is required. This is required during aerobic respiration. Specifically, you can say it is required for Krebs cycle, if you know what it is. Okay. So this is required during aerobic respiration or Krebs cycle. Not only that, you can say this is the only enzyme which is required in Krebs cycle, but which is membrane bound in nature. So, right, only membrane bound enzyme of Krebs cycle. Okay, very important points, guys. So please keep these in mind. So we are going to talk about its location altogether, where exactly it is present in the inner membrane. So we see that if this is the inner membrane, so this portion which is present outside the inner membrane, so this is the outer compartment. And the portion which is present at the inner side of inner membrane, that is your the side of matrix, the matrix side. So we see that SDH, or succinate dehydrogenase, in short forms we call it sometimes SDH. So we see that they are present at the inner side of inner membrane. Okay, so this is their location. So please write it down. They are present at the inner side of inner membrane. Okay, it's a very important point, guys, in terms of the NEET examination. You need to know, sometimes they ask fill in the blanks question from here. So this two inner side, this two inner word is very important. Inner side of inner membrane means at the side of matrix. So this is the first membrane bound enzyme what we are discussing. The next inner membrane bound enzyme we are going to talk about, so this is also very famous. I guess a lot of people know this by now. This is called F0 F1 particles. Right? You guys are shaking head, right? So F0 F1 particles, they are also known as oxysomes. They're also known as FM knob. So the word FM knob is coming because these particles are discovered by Fernandez and Moran. So by their name, they are also known as Fernandez-Moran's particle. Sometimes people call it FM knob. So if we talk about these F0, F1 particles, we see that they have three units. They have a head, they have a head portion, they have a stalk, and they have a base. Now we see that the stalk and the base together it forms the F0 part. On the other hand, the head portion, they form the F1 part. Okay. So what we see here is that F0 portion is a proton tunnel. This is a proton tunnel. And the F1 part, what we see, this is your ATP synthetase. Okay, so this is your ATP synthetase. So if we now talk about their location on the inner membrane, we see very surprisingly that this F0 part, okay, is transmembrane in nature. Okay, so this F0 portion, which is the proton tunnel, this is transmembrane. On the other hand, this ATP synthase part, this is present towards matrix. So very important informations, Often they ask questions from here. So please be very careful with it. Look at it very carefully. And if you have to ask me anything, please go ahead and ask. Can be a little tricky sometimes to understand. Now, if we see the inner membrane, we see that these membrane bound enzymes, which is succinate dehydrogenase, which is F0, F1 particle, and etc. So these are all on membrane. We will gonna talk about what is present inside the matrix, and to do that, before we do that, rather, we are going to talk a little bit about mitochondria in general. So what we have started with, right? So we see that mitochondria can be visualized under microscope by staining with genus green. So this is the name of the stain what you have to remember, which helps to stain mitochondria, okay, to visualize them under microscope. If you are using, of course, if you are using a light microscope, if you are using an electron microscope, you do not need that. But again, if you use a, this we know, if we use a electron microscope, the mitochondria will be dead. So there is no, live functioning what you can visualize with an electron microscope. Obviously, you get a better resolution. Now, talking about that mitochondria in general, we see that it is having a sausage shape. Okay, I guess a lot of people, those who have eaten sausage in their home, they know how does it look like. It's having a shape of a sausage. And if we talk about the length altogether, so we see that it is having a 0.7 to 1.2 micron in length. Okay. So that we have to keep in mind. There is one more information what we need to know with them is regarding the numbers of mitochondria per cell. Now it is extremely variable. It depends upon whether or not the cell is using a lot of energy, because we know this, we have studied in our lower grade 9th or 10th that mitochondria is known as the powerhouse of the cell. They are important to produce ATP. So we see that if a cell is involved in a process where it requires a lot of energy, the number of mitochondria will be more in that cell. So look here, as per the need of energy, the number of mitochondria vary per cell, right? So if we require more energy, the cell will have more mitochondria. If we require less energy, the cell will have less number of mitochondria. Now going ahead with that, we see that mitochondria can also take part in binary fission, or you can say the binary division. So if the mitochondria has to increase their number inside the cell, they will go under the process of binary division. So we see that, let's imagine this is a mitochondria. So this mitochondria, if it needs to divide, it will directly, take the path of binary division. And from there, just like bacteria, how bacteria take part in binary division, it also going to create two of the daughter mitochondria here by the help of binary division. So you see that the inner membrane which is enfolded to a lot of places to increase the surface area. While discussing about this, we see that mitochondria in the membrane section, where we have discussed that the inner membrane of mitochondria, where it carries a lot of proteins which helps them to, in different metabolic processes, what we have forgotten to mention that the inner membrane of mitochondria, the inner membrane of mitochondria carry a prokaryotic lipid and this lipid, this is known as cardiolipin. So this presence of cardiolipin, it is an indirect proof. Okay, it's an indirect proof of, this is an indirect proof of endosymbiotic theory. Okay, so enough about membrane, right? So we have talked a lot about membrane. So let us now talk about the matrix. So we see that matrix is a busy zone. Mitochondrial matrix, mitochondrial matrix, there are several metabolic functions which are happening in this area. So if we try to understand what are the things which are happening in mitochondrial matrix, we perhaps have to look inside it. So we see that mitochondrial matrix, okay, this is the site for protein synthesis. So you see that here you can see protein synthesis, site of protein synthesis. You know there is the presence of 70S ribosome, which are often come and organize themselves as polysome, right? All of these things you can see. This we have mentioned already. This is the site of aerobic respiration. This is the site of aerobic respiration, right? So here you see two of the very important process here, which are known as link reaction and also the very famous Krebs cycle. Beside this, we see that mitochondria, this is also mitochondrial matrix actually, this is also the site for fat metabolism. So you see a specific event which is known as the beta oxidation of fat, which is happening inside the mitochondrial matrix. So we see that the fatty acids are getting oxidized to produce acetyl-CoA by this process, and that acetyl-CoA we will see later on also take part in the process of Krebs cycle. So all of these separate metabolic functions are happening inside the mitochondrial matrix. So as a fun term, we can call it the metabolic hub of cell. This is the metabolic hub of cell. So a lot of metabolic functions are taking place here, right? Now, obviously, we forgot to mention about major three functions. So you see, one I have mentioned, the protein synthesis, the replication of their own DNA, transcription, all of these are happening inside the mitochondrial matrix. If you want, please go ahead and add them. Replication of their own DNA, and RNA synthesis as well. Okay, so all of these we can see in mitochondrial matrix. Now we see that mitochondria, this is known as the powerhouse of the cell. So it is known as the powerhouse of the cell because this particular cell organelle is very famous for the synthesis of ATP, right? So we'll talk a little bit about that. So we see mitochondria the powerhouse of the cell. So we see that the process of synthesizing ATP in mitochondria, this is known as oxidative phosphorylation. Okay, so this we know that ATP synthesis altogether is known as phosphorylation, and in mitochondria this process is known as oxidative phosphorylation, right? By the help of oxidative phosphorylation, by the help of oxidative phosphorylation, mitochondria produce ATP in large scale. Now, this process of oxidative phosphorylation, it does not run generally alone. It run along with another process which is called electron transport chain. So we are going to discuss in detail about electron transport chain in the chapter of respiration in plants. But here, what we have to know a little bit about electron transport chain is where exactly it is happening. So we see that oxidative phosphorylation is a coupled process. It's a coupled process with electron transport. Sometimes you call it electron transport system, or we call it ETS. Okay. So we see that the electron transport system, this process is specifically So this process is specifically happening in the inner mitochondrial membrane. So if we talk about the site is inner mitochondrial membrane, right? But now if we talk about oxidative phosphorylation, we see oxidative phosphorylation, the site is both inner mitochondrial membrane and perimitochondrial space. So inner mitochondrial membrane and perimitochondrial space together, it helps in the process of oxidative phosphorylation. Now, this is almost what we need to know about mitochondria, and we are going to conclude that with the last topic regarding mitochondria is that mitochondria can take part in a process called maternal inheritance. So now we are going to discuss about maternal inheritance. Now, this is something again a topic for discussion regarding class 12th syllabus, which is the principles of inheritance and variation. There we're supposed to discuss this, but generally what we see is maternal inheritance is a cytoplasmic inheritance. So a cytoplasmic inheritance is coming from the egg only. So we are going to have a quick look in the process of fertilization. So if we talk about fertilization, we see in the process of fertilization, there is an egg which is carrying the genetic material, right? So this we know. And there is a cytoplasm of egg. Now the cytoplasm of egg, it carries mitochondria. It carries the mitochondria. It also carries the chloroplasts. If we are talking about a plant cell, right? So it can also carry the chloroplast. And it can also carry mitochondria. Now, we see that if a sperm is coming for the purpose of fertilization, here we see that the sperm, I'm trying to draw a basic diagram here. So the sperm here is only donating in the process of fertilization, it is only donating its nucleus. So if the fertilization takes place, we see there will be the production of zygote. Now inside the zygote, there will be the mixing of genetic material. So you see that this is the mixing of genetic material of egg and sperm, right? But if we talk about the cytoplasm of the zygote, we see that cytoplasm of the zygote is carrying the mitochondria and chloroplast. So the mitochondria and chloroplast of zygote is directly coming from the egg cell. So this is something what we have to look carefully, that egg, this is the donor of mitochondria and plastid, or you can say chloroplast in this case, to the zygote. So now let's imagine we are looking at a character which is determined by the genetic material of mitochondria. Okay, so for example, let's say I have a faulty gene which is present in the genetic material of mitochondria, and because of that, let's say I cannot digest, let's say, fat, right? So if that happens, that mitochondria I have received only from my mother, because mother is donating the egg. Sperm, on the other hand, which has been donated by my father, this is not going to contribute for any mitochondria or plastid to the zygote. So we see that the mitochondria and plastid is donated to the zygote only by the female gamete or the egg cell, and hence we call it the maternal inheritance. So one of the examples of the character what we have to learn here regarding the maternal inheritance and its consequence with mitochondria is cytoplasmic male sterility in maize. So this is again a frequently asked question. What we have seen has been asked many times, that because of which cell organelle we get to see this disease in maize, which is called cytoplasmic male sterility. So this occurs due to the faulty mitochondria. So if the female parent is having that disorder, that particular disorder in their mitochondria, obviously all of the offspring is going to carry that disorder, because the female parent here is the donor of mitochondria to the next generation or to the zygote. Okay, so that is all about today's discussion regarding mitochondria. We will catch up soon on the discussion of plastid. Okay, tata and take care.